welcome to my channel Sam Biology Class. Friends, today we will discuss about zinc finger nuclease. In addition, we will also come to know that how this targeted nuclease is used for genome editing. But before that, will you please consider subscribing my YouTube channel? Your subscription is often a lot to me. Now the question is, what is zinc finger nuclease, ZFN? It is an artificial restriction enzyme that is used as molecular scissors to cut the DNA at specific location. Other targeted nucleases such as CRISPR, Cas9 and TALEN also do the same job. Zinc finger nucleases are generated by fusing a zinc finger DNA binding domain with a FOC1 nuclease domain. The function of FOC1 nuclease domain is to cut the DNA. On the other hand, it is the function of zinc finger domain to bind the DNA at specific sequence. So by binding to DNA at specific sequence, zinc finger nuclease enable the nuclease domain to cut the DNA at targeted site. This cleavage enables targeted genome editing, which is useful in biotechnology research and potential gene therapy. This figure illustrates the interaction of a pair of zinc finger nuclease with one double-stranded DNA. As already said, each molecule of zinc finger nuclease consists of a FOC1 nuclease domain and a zinc finger DNA binding domain. Nuclease domain cut the DNA. On the other hand, the zinc finger domain binds to DNA at specific sequence. Each zinc finger domain consists of a series of zinc finger motifs. In this figure, the DNA binding domain is composed of 4 zinc finger motifs. All of them together form a zinc finger array. Each zinc finger consists of an alpha helix and an antiparallel beta sheet. The beta sheet consists of 2 strands. In addition, each finger motif is also associated with zinc ions. This DNA binding domain is connected with the FOC1 domain by a linker sequence. Each zinc finger motif recognizes and binds 3 nitrogenous bases of DNA. As in this domain we have 4 zinc finger motifs, so together they recognize and binds to a sequence of 12 nitrogenous bases of DNA. In order to cut the DNA, FOC1 nuclease domain must dimerize. So by recognizing and binding to the specific sequence of DNA at appropriate location with the help of zinc finger motifs, 2 FOC1 nuclease domain come together and form a dimer, which then creates a double-stranded break. This double-stranded break at specific location enables targeted genome editing. An important point is that the DNA binding domain is customizable. That means scientists can design individual zinc finger motif in a specific manner so that it will recognize and binds to a short sequence of 3 bases. After designing individual motifs, they are fused together to form the zinc finger DNA binding domain. In next step, the DNA binding domains are combined with nuclease domains to generate molecules of zinc finger nuclease which will cut the genome at a location that the scientist desires. Zinc finger nucleases are useful to manipulate the genomes of many plants and animals and also various types of mammalian cells. Now the question is, what is genome? It is the haploid set of chromosomes in a gamete or microorganism or in each cell of the multicellular organism. Genome editing is a set of technologies that is used to precisely add, remove or alter DNA within the genome of a living organisms. Chromosomes are the part of the genome. So for genome editing, we have to make necessary modifications in the target chromosome. For genome editing, first of all, we have to create double-stranded break at the specific location of the chromosome by using targeted nuclease. Zinc finger nuclease is one of the tools of genome editing. In this example, a dimer of zinc finger nuclease has been used to produce a double-stranded break. Consider that this is the part of a chromosome and this is a specific gene. So this pair of zinc finger nucleates which binds to specific DNA sequence produce a double-stranded breaks which will be repaired by cell's natural DNA repair machinery using two different mechanisms. First one is non-homologous and joining NHEJ, and second one is homology directed repair. Non-homologous and joining directly ligates the broken ends of DNA. It doesn't require homologous DNA template. On the other hand, homology directed repair requires a homologous DNA template. In this figure, this is the double-stranded break. Zinc finger nucleases can be used to disable dominant mutations in heterozygous individuals by producing double-stranded break in the DNA in the mutant allele, which in the absence of homologous template will be repaired by non-homologous end joining. Non-homologous end joining directly ligates the broken ends of DNA, usually without causing any mutation, provided the cut is clean and perfect. But when the repair is imperfect, this can lead to insertion or removal of few nucleotides from the target site. So by doing so, a mutant allele can be disabled or it can be knocked out. Other mechanism is homology directed repair that invokes the process of homologous recombination. Homologous recombination can be used to rewrite the sequence of an allele by using homologous DNA template. In homology directed repair, a homologous DNA sequence is used to repair the double-stranded break by homologous recombination that can lead to targeted gene replacement. Another application of genome editing is gene therapy. The success of gene therapy depends on the efficient insertion of the therapeutic gene at the target site within the chromosome without causing cell injury, oncogenic mutation, or an immune response. Friends, this is all about zinc finger nuclease and how it is used in genome editing. For more updates, subscribe my YouTube channel. some biology class.